So hi, hello and welcome again. Microbehunter here, and welcome to another Saturday Microscopy live stream. Today it's at a slightly different time. It's a little bit earlier, about 7 hours earlier than normally, because I have an appointment and therefore I could not fit it in. Otherwise, by the hope that you were still able to join. If not, then of course you have the opportunity to look at this live stream later on. I'm going to, every time I'm going to quickly check if everything works or not. Okay, I can see myself that I'm online. If you've joined as well, I would like to ask you please to quickly type into the chat if you're able to hear me properly. Because so far I do not see that anyone is typing anything. Yes, hello, hi. Yes, the chat also seems to work. And maybe we're going to have a few new listeners today. I don't know. It's because we've got viewers from all over the world. So because of the time difference, it could be that the time right now is a little bit more or less appropriate than before. So yes, it seems to be working. Hello, everyone. Thank you very much for tuning in again. Okay, I will be doing the following today. I've kept the topic a little bit open, but I do want to show you, again, a few people have sent me their home lab, basically a picture of their microscopy setup at home. I'm going to show you 3 pictures there. I'm also going to show you 2 interesting mobile phone adapters, which I bought. We're going to have, they're a little bit different, a little bit unique, so you might be interested in that. And I'll of course also be answering any other questions that you might have. Please write them into the chat, okay? Yes. So there are already a few things here. Yes, I can help you. Okay, hi, please name my name, okay? Yes, hello from Tennessee. Yes, it's been always a tradition in the past couple of live streams that if you want to, you can tell me from where you are. You can type it into the chat because then we can see that from wherever the people are from all over the world. So so what I will be doing is the following. I received a few pictures again. If you also want to send me your pictures from your home microscopy setup, you're welcome to do that. But this is the first picture that I got. Okay. Quite interesting. Some of the home labs are quite elaborate. Others are, less so. It depends always a little bit in what your microscopy interests are. I consider this one as actually probably one of the more elaborate ones. What we can see is here on the left. Let me see if I'm able to point here a little bit. Here, the pointer works. This seems to be here a centrifuge. I've also got a centrifuge. Of course, then a regular compound microscope, another compound microscope over here, a stereo microscope. And then, of course, a whole bunch of lab equipment, parafilm. Yeah, that is a very common one that I've also used quite a bit. Okay, so yes, hello from Tennessee, from Bulgaria, Ohio, from Sri Lanka, in Sweden. Okay, nice and cold. Yes, from China. Okay, welcome from all over the world. It could be that today a few other people are also tuning in because, as I mentioned already at the beginning, the time is a little bit different. Normally I do the live stream at around 9:30 in the evening at my time in Central Europe. And this could of course mean that some people are not able to see that. So yeah. Okay, from the Philippines, from Australia. Okay. So, Avi from Romania, yes, okay, so, so this is basically the first picture. If you also want to share some of your microscopy lab pictures, you can please also send them to me. My email address is the following. I'm going to go over here if you want to send me your, Oliver at Microbehunter, Microbehunter.com. Okay, so if you want to send me pictures, you can send them to me, and I'll also put them then into one of the live streams. So and another picture that I would like to show you over here is another one that I got. Okay, this one also is the view from the back and the view from the front. Here as well, we can see that there is a a compound microscope over here, and over here also a stereo microscope in the back. We see the arrow first. Here's the arrow. Okay, yeah. So you see that there are quite a few different interesting laboratory setups that there are. Okay, so yeah. Hello from China. Okay, from the Philippines, Australia, Vietnam, Tennessee. Yeah, because of okay. Yeah. So a few minutes left. Yeah, from Kashmir, also from Vietnam. Okay. So yes, so what I would like to do in the first couple of minutes is I would like to show you a couple of mobile phone adapters that I bought myself, and they're quite interesting. They have advantages and disadvantages, as so often, but because they're a little bit unusual, I thought I'm just going to share them with you. Basically, this one over here, I can also show you the original box here. This is how it looks like. Okay. I got this one just recently. I did not buy it over Amazon, but over a shop. The company actually offered to send me the product for free, but it was not so expensive, so I simply bought it online. That's basically the brand name, and it's quite an interesting mobile phone adapter for microscopes. I want to show this to you. I have to take out the eyepiece from the other one to demonstrate this. Okay, up here it is. And it works like this, that if you're able to press this together here, then you see it works like a clamp. There are some foam cushions over here that you had to glue in. And then basically the eyepiece of the microscope goes in like this. Okay, and it's held in place. Okay, and over here, this is of course the place where the mobile phone is connected. The camera lens should be here, and you can see that you can shift it left and right. And it works a little bit like this, that here there is the camera lenses over here, and then you have to put it in like this. Okay, you have to make sure, I'm just going to show you here first, you have to make sure that this is all centered, the camera. Okay, you have to shift it. Okay, and then basically it goes on top of the microscope. Okay, and you can do then microscopy like this. This is a very interesting concept. Okay. Some advantages and disadvantages, of course. One of the nice things is you can of course also connect a tripod here, so if you need additional stability, and then you can mount it there. The advantages are it was not too expensive. That's a good thing. I think it cost around 17 euros or so that I paid $17-18 if I remember correctly. However, there are a couple of things that you simply have to be aware of, and it depends a little bit on the mobile phone that you have, whether it's a suitable one or not. Number one, as you can see, it's not all the way in, obviously, because in this case, the camera lens is quite far on the top, and also when in my other mobile phone, the camera is very much to the side. Okay, so you have to make sure that then it has to go all the way to the side, and if it does not work, then you have to flip the camera around, and then move it to the side. The disadvantage here is of course, as you can see, that the clamp here kind of covers up some of the buttons. Okay, but I did check, and luckily, and maybe the clamp does not push the buttons, because the clamp kind of holds it from the top, and for this reason the buttons are not pushed, so that's an advantage. On the other hand, the disadvantage is a little bit, you see, especially if the camera is very much on the top, like this over here, it's still a question of stability. Okay, but it's an interesting concept, and I would say that it's worth trying out. It depends a little bit on the size of your mobile phone and the position of the buttons and everything, of whether this is actually something which to the camera is basically appropriate. Okay, and I found out that especially with those eyepieces that I've got over here, it's too far away from the camera lens. Okay, so the eye relief of these eyepieces here is too short, so that means even if you put it all the way in, you only get a smaller circle, and it's a little bit blurry on the edges because I cannot get it closer. Okay, so this is a little bit a disadvantage. So you see the whether or not this adapter will work for you or not depends actually not only on the adapter itself, but also on the size of the mobile phone, on the type of eyepieces that you have. Okay, but I thought it was simply still an interesting concept. Okay, especially easy way of connecting it and disconnecting it is something that I kind of like here. Also, if maybe the mobile phone is a little bit too heavy, I mean, it doesn't slip out, yeah, but yeah, I don't know, very expensive mobile phones, you got to be careful that it doesn't fall out, okay? But actually, it's relatively stable, okay? But then again, over here as well, it's just the spring that you have over here that kind of holds the eyepiece, okay, in place. So, yeah, that's simply something I just wanted to make you aware that this is essentially the way that it works here. I'm going to read again the comments over here, okay? It's night time here. I have a few minutes left, okay, from Kashmir, okay, from Vietnam, Germany, okay. Yes, it's amazing to see you live. You're one of my favorite channels. Thank you very much, okay. Why don't you show any specimen preparation nowadays? Okay, I can do that if you want to, okay. Video on gram staining. Oh, gram staining, that's an issue, okay. It's a little bit elaborate, I think. Okay, this is my first live with you, so I'm excited. Okay, yeah, usually, as I mentioned before, I do the live streams a little bit at a different time, but today I had to, because of an appointment, I had to move, to kind of, shift it around a little bit. But you know what I'll do? Thank you for the recommendation, and I'll do some specimen preparation the next time. Okay, and the CD fuge. Okay, I've been working on this a little bit. I'm still trying to optimize the design a little bit. Okay, the CD fuge, that is a centrifuge which uses a CD and strings to pull. But I want to make sure that this works fine first before I demonstrate it to you. Okay, so this is basically the one that I wanted to show you, and here the other one that I've got over here is quite an elaborate one. It's actually from the company Celestron. They are making telescopes. It's actually for telescopes, but they also included microscope adapters, so they're selling it for telescopes and for microscopes. And look at this. This is a complex design. Okay, for a mobile phone, and I'll just quickly show it to you. What you have to do is the mobile phone, of course, goes in here. So what you have to do is you have to pull it. Okay, then it goes all the way in here. It goes all the way to the bottom. Luckily, over here there is an opening so that you can plug in any cables that you want. And then there are basically 3 axes that you can control. So first of all, you can press this button, this lever over here, to hold the eyepiece. Okay, and you see that it's basically too large, and for this reason you need to add this adapter, which was also supplied. Kind of just put it over. Okay, like this. Yeah. And then what you do is, let me move the mobile phone out of the way again a little bit. Let me take it out again. And then what you do is you basically open the clamp by pushing this in. It goes in here, okay? And then what you can do next is you can turn this screw over here to tighten it, okay? And this way, it's pretty tight. And you're also not able to open it anymore, okay? So this is a little bit the advantage of this system, that it's actually significantly tighter than, for example, this one over here, yeah? So this is actually quite nice. Of course, look at this. This still slips in and out a little bit. So yeah, I guess it depends a little bit how hard you clamp it together. So, but the interesting thing that you have here now is the following. There are 3 knobs that you can turn. This one here looks like a little bit like a mechanical stage. And then here, this one over here. And look what it does. If you turn this here, yeah, it moves. Maybe you see it better over here like this. Yeah, it moves the phone horizontally. This one over here, yeah, moves that's the y-axis. Yeah, and this one over here allows you to adjust the height. So this allows you to completely align the camera of the mobile phone with the eyepiece. Okay, so, and there are additional screws. I did not try those, but apparently those tiny screws that you can use here, is if you tighten them, then you can actually fix it and you can completely clamp it so that you cannot turn it anymore. So kind of they allow you to adjust the friction. Okay, so this is basically the system over here. One thing that it does not allow you to adjust, maybe also not necessary, is the tilt. Okay, so you see there is a little bit of a tolerance here as well. Okay, so maybe by adjusting some of the screws, you're able to deal with that a little bit better. Yeah, it's much more expensive. I think I paid around 70 euros, which is around 70 US dollars for this. I think it is useful, especially then, if you have to exchange the mobile phone frequently, especially maybe you're in educational setting where you are together with students, where each student should use basically has a different phone, and you want to quickly align it so that every student is able to take pictures. Then I think this one is kind of suitable. There is, however, one thing that I want to also tell you, especially if the mobile phone is too small. I don't have a small one with me right now, but if it is too small, then it's not able to go all the way down. So basically I had a small one, mobile phone, and it wasn't basically the camera. To get it aligned, it was not able to go all the way down, okay, because the mobile phone was too small. Yeah? So that is essentially something that you have to be aware of. And another thing that you have to be a little bit aware of, which I think is a little bit of a pity, especially when the mobile phone is small. Look, if you move it all the way forward like this, then you're not able to lower it completely because it blocks here, obviously. Yeah? So this is a question of the size of the mobile phone that you use. Yeah? Yeah. Otherwise, it's an interesting idea. It's a little bit bulky, I have to say. Yeah, but just wanted to share it with you. This idea. Okay, so I'm going to read a couple of comments again. Fungi mycelium smash mounts would be awesome. Okay, so I'll do some do some squash mounts. Hello. I got here. I would like to see more amoeba videos. Seriously? Wow. Okay. Time lapse. What microscopes do you own? A BX50? No, a BX53. Okay. Hydras are interesting too. With the hydras, I've made some videos, but I don't have any right now to look at. So I have to go out to the pond and maybe I can catch a few more. It's relatively warm right now in Central Europe, so it shouldn't be a problem. Okay. Ah, yeah, you have the Celestron Adapter. Okay. Did you get my lab pictures? I did get some lab pictures, but I showed them already last time. I don't know if these are the ones that I showed you. This one is especially useful when you want to take photo or video through the wide field eyepiece. Okay, it's a little bit heavy. I mean, let me show you some other lab pictures. These are mine. I've done some away. I don't know if they're right now here. I'd have to check again if I've got them because I changed some of the lab pictures around. So I hope that I got them. Okay. So what I would like to do next. So that's basically just two adapters I wanted to show you. If you have any questions about anything microscopy related or specimen preparation related, yeah, please feel free to ask. So otherwise, I'm going to move on. There is a couple of questions that I receive every time, and it's a very common question, but I simply wanted to illustrate this a little bit. Is this the question about the blue background of some of my my videos that I'm making? And you know what? Maybe this is a good opportunity now to do a little bit of preparation. Just a second. Okay, why not? I've got over here this so-called hay infusion. Let's see if we're able to find a few few ciliates here, and then I'm going to show you a little bit the settings of my microscope, because some people wanted to know that a little bit, and maybe this is now also a little bit the preparation. And then later on I'm going to show you if I don't forget about it, I'm going to also show you the unfortunately, it didn't work, so I have to send it back. I bought myself an ultrasound cleaning device that I wanted to use to clean my microscope slides. Okay, so yeah, what I do is occasionally I use those large cover glasses because those large cover glasses do have the advantage that the specimen does not dry out so quickly. Okay, but unfortunately, like so often, when you buy them, they are a little bit greasy and dirty, and then I have to dry wipe it. I'm doing this generally. The microscope slides here, I have already washed them, so they are reasonably clean already. Okay, so there's no need to dry wipe them. I'm kind of trying to reuse it a little bit. And then let's put a hay infusion on here. So what is this, and how does this actually work? There is a little bit of dry grass in here, and then you add pond water. That's important, not tap water, pond water. Because in pond water you have a lot of interesting microorganisms which are not present in tap water. And also maybe if you add a little bit of the sediment or some slimy stuff from a biofilm from leaves or from rocks. But you add a little bit of dry grass and some water, and then you let it stand for a week. And what's going to happen is the following, and you got to be a little bit careful with this, because every time when you do stuff like this, you're basically multiplying and growing bacteria, unknown bacteria, and you don't really know what you're growing here. So be careful with these things, okay? It's important that you do not put a lid on top, because it needs oxygen, okay? And then what I do is I take some tweezers, okay, or a plastic pipette. And what you do is you simply take a small drop and you put it on here, okay, and you put a cover glass on top. Now I know that it's not the most interesting specimen, I'll admit. So now where do I put this? Because this one is dirty now. I don't want to, yeah. So I put it on here on some tissue paper, and then now I put the cover glass on top. Okay, you can see it over here, and yeah, you see that the liquid spreads, and now let's put it under the microscope. And I would like to explain to you some of the settings that I've got here. So, and maybe we're able to see a few moving organisms, or if the density is not very high, and it does not seem to be very high. Today in the morning I was a little bit more lucky, because this is not very interesting. There were plenty of them today in the morning, but maybe because the jar was standing, and therefore they collected on the bottom, and now because I touched it, I kind of agitated everything again, and now maybe the concentration is simply too low. Okay, that's kind of disappointing. Okay, yeah, but that can happen. But I know there are plenty of them here, but it could be indeed. There's a very tiny one that I see over here. But the thing is because I agitated it too much, and now everything was kind of spread all over the place. Yeah, I mean, I do see a few of them, but they're too small. You know what? We want to see something more interesting, so I'll grab a different one. Okay, so this one over here, this is water from an aquarium. Okay, this is water from an aquarium that I'm going to do. So I'm going to read again here some of the question. Is it possible to do polarized light microscopy without removing the blue filter of the microscope? Yeah, of course. All you do is you put a polarizing filter directly over the lamp. You can do that as well, or you put the polarizing filter on top of the blue filter of the microscope. Doesn't matter. Okay. Yeah, please check. I'll check again, because I do want to share your pictures with the home lab, but somehow, yeah. How do we keep water sample in our room? Okay. How do I keep those? I kept them next to the window, simply because there is space there and there is light there, but keep it out of direct sunlight. Sunlight is going to heat up everything, and then everything when it's too warm, then this will also drive out the oxygen. But a light is always good, because some of the algae in there, they will do photosynthesis and they will produce oxygen this way, and the microorganisms actually like that. Okay, okay. Yes, you warned all of us to do a hay infusion, but why did I do that? Yes. Because I warned to do hay infusions, because anytime when you do things like this, you are growing bacteria. Okay, and the thing is the following: hay infusions are a traditional way to do microscopic specimens. However, it is just like when you're spoiling food. When you're spoiling food, when you've got a sandwich that is kind of rotting, you're growing bacteria. Every time when you do things like this, you have to be careful. Okay, and especially if there are children around and you're spilling those things, you got to be careful. But I would say it's not a reason to be extremely paranoid or anything. But there is a healthy amount of care is always a good idea. Okay, and I know that especially also in schools, yeah, people are doing that, but it's just like spoiling. I know some teachers are doing that. They're buying some food, a hamburger, and they're spoiling it and watching how it rots and decomposes. It's also not good. Okay, and also here you always have to be careful. So yeah, because you're working with unknown microorganisms. Yeah. Yeah, and especially I have less problems with an hay infusion, but one of the things that I really would not do, and you do see YouTube videos where people actually do that, is they're buying agar plates and then they're making fingerprints. Basically what they're doing is they're growing the bacteria that they find on their skin. And that is something that I would really not do, because these are neat bacteria that are able to grow on your body. They're potentially pathogenic, disease causing, some staphylococci or so, and I would not grow those. Yeah, with a hay infusion and so on it's a highly complex thing, and you also don't know what you're growing. Okay, but I see I need a little bit more of this, but I would be careful in any case. Well, let's add a little bit more. Okay, so and let's have a look here. Okay, so and I will switch over to the microscope again, and I'm not happy with the first one with the hay infusion, so let's remove this. But with this one I hope to be a little bit more successful. Okay, and we see already a few things here. Okay, it takes some time for the water to properly spread. Yep, and this one over here, of course, is an air bubble, and over here we already have a paramecium. Okay, I think and a rotifer over there. I think the amount of water is a little bit much, therefore the things kind of go in and out of focus a little bit. Okay, and what I've done here is I've taken some water from an aquarium, a home aquarium, with some plants, and I simply let it stand, and the plants started to slowly decompose over time, and this is basically what I got after a week or so. Yeah, and yeah, so there are plenty of different microorganisms here, and again some questions, and I'm going to show you a couple of the settings that I've got here. Okay. Does Graham's iodine is necessary for gram staining? Yeah, you do need, there is a so-called Lugol's solution that you need for gram staining, which is an iodine solution. Okay. I also do this to my water samples, yes. So how is it's made? Will my fish pond have water organisms? Of course. Of course, this is absolutely necessary. However, if you take water from a fish pond directly, just the water, you're not going to see a lot of water microorganisms, so you have to take some sediment from the bottom. But especially from fish ponds and also aquariums, the problem is the following, that fish like to eat those. So this means that if you take a water sample with some decomposing leaves, you have to let it stand for a couple of days in your room, so that they are able to reproduce, because in nature sometimes the fish will eat them up as soon as they reproduce. Okay, so now I would like to show you a couple of settings here. So what you're seeing right now, this is known as this one is the correct setting. This is known as DIC, differential interference contrast microscopy. And what I'm able to do is by shifting a prism at the top, okay, I'm able to change the background color. Okay, so that is basically because some people ask me, how is it possible to get this blue background? And I don't know, maybe some of you are also seeing the very nice YouTube channel Journey to the Microcosmos. It's also a microscopy channel that I also like to watch a lot. Yeah, they are also having always a very nice blue background. Okay, so it's the same system. Yeah, of course, I mean, I could change it, but essentially, yeah, blue is nice because it kind of implies a little bit water. Okay, so how would everything look like under a regular bright field? I'd simply, do like this, and this is how it would look like in conventional bright field. Okay. And if you want to get color anyway, then I recommend that you use some color filters. I've also made videos. Yeah, they also work quite nicely. Okay, but there is something interesting that I'm just seeing over here. Let me put this back here. Because somebody was kind of asking me. Look, let's go up with the magnification a little bit. I have to go up with the magnification center, and it's too dark right now. I'm adjusting now the camera a little bit. Okay, and because somebody was asking me about amoeba. Okay, I'll show you now the arrow over here. You see this guy here? Okay, that's an amoeba. And you see it's slowly moving. So what I need to do now is you need to take a time-lapse video and then speed it up quite a bit. Okay, so I'm going to move up to 60 times magnification here. It's a little bit too dark. Let's open this again. And the higher you magnify, yeah, you can see how it's slowly moving around. Okay, so because somebody was asking about amoeba. So let's go over here. Safranin staining, yeah, that safranin is also a very common stain. Safranin is the so-called the counter stain for gram staining. It gives a red color. Okay, what camera are you using for this live video? I'm using now a Canon DSLR, a Canon digital camera, yeah, and that's basically. I've got a couple of Canon cameras. I like those, but other cameras will work. So I do not use a mobile phone, and I do not use a separate microscope camera. They're those USB microscope cameras, and the reason why I do not use the USB microscope cameras is not because they're bad or anything, but because the USB connection is too slow. At least the USB 2.0 connection is too slow for video. And I want to take high resolution video. So what I do is I take the video directly into the camera. That's the only reason. Okay. Yeah. Journey to the Microcosm is also one of my favorite channels. Yes. Okay. Can you make a video about bacteria living inside root nodules of legumes with your new microscope? Wow. The microscope, sure. But I have to get the preparation. That's a difficult thing. So what I have to get is I have to get some root nodules. Okay, and then I somehow have to extract the bacteria. I've never tried this from root nodules before. Okay, but that's a worthy challenge. Okay, are pocket cameras better than phone for taking images under the microscope? That's a good one. Thank you for the question. Oop, now I bumped it. Okay, so pocket cameras, I don't have one here right now, but pocket cameras have certain advantages and disadvantages as well, because I've experimented around with those as well. Pocket cameras do have the advantage that they have an optical zoom. So if you zoom in, then you have the possibility to actually get a picture, not with where the image is in a circle, but one that fills everything. So that is a nice advantage of the pocket cameras. The difficulty is connecting it to the microscope. So if you want to film with a pocket camera, I recommend that you maybe set up a separate tripod next to your microscope and get everything aligned. Okay, then you can essentially also, so if you have a pocket camera, definitely would try that first with a tripod. Okay, what all microscopes in common? What all microscopes have in common? Well, all microscopes have in common that they are making pictures of things that are too small to see with the eye. Okay, and there are many different microscopes around. The ones that I'm using in hobby, or that we're using as hobby microscopists, is of course are the light microscopes. Yeah? By the way, for me, I always consider this fascinating. This guy over here, that's a rotifer, okay? And a rotifer is a multicellular animal. It's a microanimal, so it's made of many cells. Yeah, and this one over here, that's as I just told you before, that's an amoeba changing its shape. It's also only one cell. It's pretty much almost the same size. And here, this is a paramecium, also only made of one cell. Yeah? So you see that those cells are pretty huge compared to the cells of the rotifers. Yeah, because a rotifer is made, I don't know, what, around 1,000 cells or so. Yeah. So yes, that's an amoeba. And those little extensions that you see are referred to as pseudopods. If you want to see the whole thing in bright field, it's not too bright. Okay. So this is in bright field how essentially most of you are probably going to see it. And the nice thing about the bright field is look, it looks kind of green. Why is the amoeba green? Yeah, because often it contains has swallowed up diatoms and algae and so on that actually make it look green. Okay, so what I'm just trying to say this way is that regular bright field microscopy also has certain advantages. Okay, because DIC, as I just showed you before, is essentially, it's artificial, the colors. So I also want to show you something else. I also have in here phase contrast, one phase contrast objective I have. And let's have a look at this using phase contrast. Where is this? This one over here. Yes. This is now phase contrast. Okay. You might now say this kind of doesn't look so nice. Okay. But phase contrast does have a huge advantage in that you're able to see certain structures much better that you're otherwise not able to see because they're transparent. Phase contrast converts transparent objects, especially bacteria, into gradients of darkness. So bacteria can appear dark on a brighter background. Okay. So for certain applications, phase contrast indeed is more useful. Okay, but you see, it looks quite a bit different. Yeah. So I'm saying this as well because some people say, okay, they definitely want to have phase contrast, and I say, okay, just don't have too high expectations. It really depends what you would like to observe. Okay. Yeah, I'm having mosquito larvae in my water samples. Yes, I also made already videos using mosquito larvae. However, they're pretty big actually. Yeah, so they can be a little bit difficult to observe in the sense that it's difficult to fit all of them under the microscope. Okay. Do you have only one lens in the 4x objective? I'm a little bit confused now. I mean, this here is my 4x objective that I have. You're able to see it over here. So that's my 4x objective. Okay. Yeah, and this one over here that I just showed you, that's a 40x objective for phase contrast. Okay, yeah, that's a 40x objective, and I only have one 40x phase contrast objective. Okay, yeah, the amoeba in bright field again. Okay, that is the request to put it back into bright field using a. I always have to make this a little bit, close the condenser or make it a little bit brighter. Okay. So that's again using the 40x objective. That's the amoeba. So that's the way that most of you should be able to see it. And you see that's actually quite nice. And there is another thing that I can highly recommend. I mean, I'm seeing a lot of pictures online in Reddit, for example. The Microscopy subreddit is quite nice. People post pictures there, and some of the pictures I say can be actually be made quite easily better by simply increasing the contrast. Because usually when you take a picture directly, the contrast is quite low. Pretty much all of my videos that I make for my YouTube channel, I'm improving the contrast. So people always keep on asking me, why are the pictures so good? And I says, well, actually, yeah, of course you need a nice camera and the microscope should also be nice, but everyone's got that already. That's not the limiting factor. I always improve the image quality in a video editing program or in an image editing program. All I do is I increase the contrast, and this really makes a picture sometimes appear much sharper. Okay, so that's simply something I can also recommend. Yeah, I love your videos, but I have a question: how Leeuwenhoek put water samples in his microscope? Okay, Leeuwenhoek, one of the first microscopists, what he did, that's also a ciliate. Okay, maybe possibly euplotis. I hope I pronounce it correctly. What he did is Leeuwenhoek had a single lens microscope, and if you are interested in what he was able to see, there is you can buy the so-called fold scope. Some of you might already know it. It's a paper microscope. I also made a video a few years ago, and it's a single lens microscope, and what he did is he simply put this lens in front of his eye, and on a tiny needle he put the sample. So he was actually not using microscope slides, but he kind of pinned whatever it is that he wanted to look at, or maybe a water droplet on the tip of a needle, and then he held it in front of his eye. Yeah. So the microscope slides were invented only later. Okay. In one of your videos of DNA, you say it's not really DNA, so what is it? Well, I made a video on DNA. It's actually also one of the more popular videos of my other channel. And normally DNA is way too thin to be seen under the microscope. Okay, you need even with an electron microscope, it's difficult to see. But still, after I extracted DNA, I was still able to see those string-like DNA-like structures. So what is it? And what I think it is that there were many DNA pieces together, and also that there were proteins, of which there are a lot in the cells, sticking to the DNA. So the string that you see was not actually the DNA itself, but it's almost like on a necklace. There were proteins on top of it, and that's why I was able to see them. Okay, so I think that is the reason why it was possible, because the DNA itself is way too thin and far below the resolution limit of the microscope. Okay, yeah, here the amoeba is again moving around. Okay, yeah, and if we look carefully, then we're able to also see it. Sometimes I was able to, you can see how it's trying to engulf things, and if you look into the amoeba again, you can see all of the things that it was kind of eating up. Okay. Did you get DIC by an upgrade to your current microscope, or did you have to get yes, I had to get a new one. Okay, I had to get everything new, basically because upgrading, some people upgrading the microscopes, some microscopes allow you to do that, but sometimes it's not worth it. Okay, so I've talked some years ago to someone who was actually upgrading his microscope over eBay. He bought the parts together. You have to really know a little bit about which parts fit together. And then he was able to make his own DIC microscope. But I recommend that before you go into that direction, that you first play around with filters. Because with oblique illumination and color filters, you can really get a lot. Okay, yeah. So I've seen sometimes pictures where I was not able to see, is it actually DIC or is this some kind of oblique illumination? Okay, so let me go back again. Yeah. So here again we have to open it up, so that's back again to DIC. It's very difficult to use microscope in winter because of fog coming from our. Okay, and so is this fog coming and depositing on the eyepiece? Fog from our, because of fog coming from our moth. And this is on, I mean, okay, are you talking about fog forming on the eyepieces, like water condensing on the eyepieces? If this is the case, well, then this could be, yeah, then you have to make sure that the microscope does not become too cold. So too strong temperature changes are not good for the microscope because water is able to condense, yeah? I'm not talking about how many objectives you have. The question is how many lenses inside. Oh, I see. How many lenses there inside the 4x objective? I don't know. Honestly, usually those objectives, they have a whole series of several lenses, of course, to correct all of the aberrations and the lens errors. But honestly, I don't know. I have not taken it apart, and I am a little bit afraid of taking it apart, because every time when you take something apart, my room is not entirely dust free, and for this reason, there is dust can enter it. Yeah, sorry, I'd have to check. Yeah, here again, a rotifer here on the side. Yeah. Which objective do you use to find the amoeba? Okay, right now I've got a 60x objective. Okay, but concerning the size, you have to always understand that the magnification that you get with the objective is not always the same as the magnification that you see on screen, because it depends on the field of view that the camera is able to catch. Okay, so right now I have a very large field of view, so it could be that if you use let's say a 40x objective, like right now, okay, and if I only take if the camera only picks up a small part, okay, then it will appear to be larger on the screen. Yeah, so you see that the magnification, as you see right now on screen, depends also on the size of the image that the camera actually captures. Okay. Yeah. Yep. What is your favorite? My favorite water organism, I had some difficulty to find, is called Lacrimaria. It's I only saw it 2 or 3 times. I was not so lucky so far. Lacrimaria is a ciliate, and it has an extremely long neck, and it's able to hunt around, yeah, and with its long neck, so extremely stretchable neck. It looks pretty fascinating. Okay, so I'm always happy when I find one of those. Lacrimaria is its name. And it's a pretty aggressive hunter. The microorganisms have a great ability to survive in harsh conditions. So why don't multicellular organisms adapt? So it's a little bit out of topic. It's a biology question. Basically, the question is the following, and I do get similar questions quite frequently. Is a little bit microorganisms are able to survive. Bacteria are able to survive quite well. And why are we not also able to do that? Well, or not we, multicellular organisms. And I will be honest with you, some of them are able to do that. One of those traditional ones is the tardigrades. The water bears are able to survive cold quite well. Not heat so much, but cold. And the answer that I'm going to give you is correct, but it might not satisfy you very much. The reason why we humans or other animals are not able to survive very extreme conditions is because it's not necessary. Okay, our environment, we are adapted to our environment, and we're adapted well enough that we're able to survive. Okay, and the same with the other. I mean, I could say differently. I could say, well, some of those water microorganisms that we over here are not able to survive dryness, for example, yeah, because then they dry up and then they die, and they won't come back to life because they are not able to form cysts. Yeah, so, but for whatever their environment is, it's good enough for their survival. You always have to ask the question. I mean, when some of my students ask me these questions, I always say it's actually, I've only got 2 eyes. Would it not be much better if we humans also had 2 eyes in the back? Then we could all actually see much better, right? We would have an advantage. Yeah, if we could also see, I don't know, 360 degrees. So why did we not evolve eyes also in the back? And the short answer is because 2 eyes are enough for the survival of our species. Yeah, for our environment and for our context, it works the way it does. Okay, and it's actually quite an interesting question, which it deals actually goes into the direction of evolution as well. You discussed 2 different camera phone mounts earlier. Can you show your current camera mount setup you're using? Oh my gosh, I have to basically lift the camera up. Can you see it in the corner? That's my camera. Okay, it's a DSLR, and over here on the side I have a USB connector to the microscope, so that I can do the video right now that you see. And I've also got an HDMI connector to my monitor directly, so that I'm able to see it on the monitor without having to switch on my computer. Okay, so that's simply a Canon DSLR camera connected directly to the microscope. Yeah, yeah. So that is kind of the way that yeah. Is it nighttime now? Is it time for me to go to bed? Yes, okay. Thank you for still having visited. Okay. I'm satisfied with your answer. You indeed, okay. Great that happy that I was able to answer it. Yeah. The other way of answering it is a little bit if it were not enough, basically, if 2 eyes in the back were indeed a problem, not having 2 eyes in the back indeed were a problem, okay, then I wouldn't be here right now, okay? So that's a little bit turning the question the other way around, okay? We are here because the way that we are right now is good enough for the survival of the species, and we can say the same thing about all of the other microorganisms that we have as well, yeah? So this is I don't know, is this guy trying to engulf this huge green algae over here? I don't know. It's trying to maybe engulf it. Sometimes it tries to do that, it discovers it's too big, and then it goes away again, okay? Yeah, so this is, what time is it now? Okay, it's 50 minutes. So this is basically simply a few of the things I wanted to show you. Some people also ask me about the so-called oil immersion objectives. I do have a 100 times oil immersion objective over here, which I rarely use, very rarely, because the specimen has to be quite thin. Okay, for yeah, otherwise it's going to go in and out of focus very quickly. And generally, I'm quite happy with the 40 times objective. That's also actually one of the most common questions I receive. People want to know what magnification am I using, and a very common request, and I almost never follow up with that, is where people ask me, well, can you not put the magnification somewhere in the corner of the video that you're making? Yeah, and the problem that I have is actually, it doesn't make a lot of sense, because if I basically say now, what is the magnification of this amoeba right now? Well, I can tell you that I'm using the 40 times objective, sure, but you see, it's not 40 times larger. This amoeba, it depends, the size of this amoeba right now depends on how large your computer monitor is. Because there are two different types of magnifications. One is the magnification on screen or on paper, yeah, and the other one is the magnification as magnified by the microscope objective. Yeah, so when I look through the 40 times objective and the 10 times eyepiece, I get a total magnification of 400 times. Okay, but the magnification over here of this amoeba is probably much larger than 400 times. Because we call it magnification as well, but actually it means something different. Yeah? And for this reason, the only thing that really makes sense is to include a scale bar in the corner. And so why don't I include a scale bar? And the reason is because what I do very often when I make videos is that I zoom in. So normally what I would do is I would take a video like this, and then in the video editing program I would actually then only I would crop it down so that the whole amoeba would fill the whole space. This is something I would do, but then again the scale bar won't match. So I would have to actually calculate a separate scale bar for every image or every video that I make, and it's simply too much work. Okay, that's actually one of the reasons why I do not include a magnification. But I can tell you that essentially I'm making pretty much all of the videos up to a maximum magnification of, let's say, around with a 60 times objective. Okay. Okay, where can I get a high concentration of diatoms? What you might do is the following. You go to try the following. This might take time. Yes, you can scratch some material, some biofilm off the surface of rocks and leaves, but what you can try as well is if you have a water sample somewhere, you know what, why not, seen from desk, okay? Something I could try this as well. You take a water sample with pond water, and then what you do is you simply put a microscope slide into the pond water sample for a couple of weeks. What will form is a so-called a biofilm. So microorganisms and also algae and also diatoms will start to grow on the glass. And then you take it out and you put it under the microscope. So this is one way of how you can grow microorganisms on a slide, including diatoms. But I would say that the best way to find diatoms is, and that's where I found the highest concentration, is to find a pond somewhere where there's green stuff floating on the surface. I'll show you. Okay. Something, yeah, I've got a jar here. Yeah, some green, and sometimes there are a lot of diatoms in there or growing on the side of the glass. Okay, so this is because not all diatoms, but many of them, they like to grow on a surface. Okay. I always wondered how scientists discovered microorganisms and what was their reaction. Oh, I tell you, there is a book that I can highly recommend. The book is, I think, from the year 1927. Okay, and it is called, coincidentally, I didn't know about this book. The book is called Microbehunters. Okay, the guy who wrote it is called Kuif, I think. Let me quickly go online. I'm now online. I'll simply check this now. Amazon. Okay. Let me quickly check. Yes, it's called Microbehunters. The guy's name is called Paul de Cruyff. Paul de Cruyff. Okay, it's called Microbehunters by Paul de Cruyff. You know what? I can actually type it into the message here. Paul de Cruyff. So that is yeah, Paul. I mistyped it. Paul de Cruyff and the Microbehunters. That is the name of the book. And it's yeah, and I highly recommend it. It is very, very readable. Okay, it's so it is yeah, and it goes all the way back to Anthony van Leeuwenhoek, the first microscopist, and it goes all the way yeah, into medical research and so on. Highly readable, so you do not need to be, and you don't have to have any scientific background to understand it. And it's a classic, and it has inspired many people to actually yeah, then later on study biology. Okay. So it will also talk about the reaction and the problems that some of the scientists had, the competition that some of the scientists had. Okay, there was lots of competition back in the 19th century. Yeah. By the way, I'm just 10 years old. Thank you for responding to me. You're welcome. This was a quite life-challenging experience. I love it. Thank you very much. Please, for this time, put the amoeba under one. Okay. Okay. So where's the amoeba? So the request is for me to put the amoeba under 1,000 times, and I have to find it first. Okay, is this the one over here? Ah, it's over here. Here it is. So the request is to do some oil immersion microscopy. Okay, you know what? Why not? I go up to 60 times first. Okay, so. And I'll show you to, okay, just a second. Let me put everything away. I need to stand up for 30 seconds to get my immersion oil. Okay, I'm back. So the request is to put the amoeba that you see over here crawling yeah, under my 60 under the 100th times oil immersion objective. Okay, so this is going to be a challenge. No, I think it's going to be fine. Okay, so look, this basically means if I now go into oil immersion microscopy with a 100 times objective, I am not able to go back down with the magnification anymore, because if I were to go back down with the magnification, I would contaminate my other objectives with immersion oil, and I cannot do that. It's okay. So once I'm in oil immersion, I have to stay in oil immersion, or I have to change the slide. Okay. So now the amoeba itself is slow enough, and it crawls along the cover glass or the microscope slide. So it is slow enough so that oil immersion actually makes sense. Okay. So let's try do that. Okay. Is fish blue as an alternative to methylene? I don't know what fish blue is, and honestly, I really don't know. I just know that some aquarium shops sell methylene blue as a treatment for their fish. So you can already see that the movement is not quite as smooth anymore, and the reason is because the light intensity, even though it's already at full maximum, is too low, so the camera is not, yeah, the camera is a little bit too slow. So this is just for your information. So, but let's everything's set up now. So what I will do now is I will now center the amoeba. Okay, and I will now. And now you have to check what I'm doing here down here in the corner. Okay, you got to check what I'm doing here. I'm now going to add immersion oil directly on the microscope slide, and then I'm going to rotate the 100th time oil immersion objectives into position. Okay, so that's why the image is going to disappear now. A drop of immersion oil goes on the slide. Okay, and the oil immersion objective goes into it. And then sometimes what I do is I go back and forth a little bit, and I don't see anything because the light is off. So, and this is now a little bit too dark. This is now, I have to adjust the camera a little bit, make it a little bit brighter. And this is now using the 100 times oil immersion objective. You see, it's a little bit out of focus. Okay, and this is how the amoeba looks like in 100 times. Let me, so I'm adjust, the reason why it's brighter, and next to it is a paramecium. Okay. Yeah. So this is basically now oil immersion. So I'm going to now show you how this looks like without DIC in regular bright field. I have to lower the light intensity. Okay, so this is now a conventional bright field, and you can see that actually for some cases the bright field is actually quite a bit more useful, because now you can see the diatoms and some other algae that the amoeba has devoured. Okay. So I put the algae under the microscope and wonder how can someone tell if it's alive or dead? Well, I mean, I can tell you the following: the algae, when they're green, and when bubbles are forming of oxygen, then they're definitely alive. Okay, oxygen bubbles are a sign that something's going on here. Yeah, so this one here is now the amoeba. You know, we have to be a little bit lucky now to find a paramecium. There are some other ciliates floating around here, but you see how quickly they are moving, and that's why it's impossible to chase them. Yeah, so that's why the high magnification oil immersion is actually only useful, yeah, for objects that are moving kind of slowly. Yeah. Does it make sense to always use more than 400 times? No, definitely not. Especially when the object is very large or fast moving, you have to use a lower magnification, and also the depth of field is much lower at a higher magnification. So there are always some parts which are out of focus. I mean, for example, right now, you can see that the bottom part here of the amoeba, yeah, is in focus, yeah, and this one over here not so much, yeah. And at a lower magnification, the depth of field is larger and much more is in focus. Yeah. So it really depends a lot. But the problem again, that I'm saying is right now I cannot switch back down to a lower magnification, because then immersion oil is going to go on my other objectives, and then it's going to be damaged, right? This actually happened to me when I was several years ago. You kind of switch down to a lower magnification, and then you have to clean the other objectives again and something, yeah. So this is a little bit the idea. I got a little bit sidetracked. Actually, I wanted to talk more about also other things here, but I think it's also quite nice to look at some amoeba, some water samples. Okay. Yeah. So I'll have a look at here. I think I read all of the comments already. Okay. Yeah, so this was basically, I can move around a little bit more. Maybe we're able to find something else. Yeah, that's a rotifer. You see, as soon as they pass through, they're immediately gone again. Okay, there seem to be some kind of diatoms here. Yep. Yeah, that's basically because there was a question about diatoms. Diatoms, here we've got one. Yeah, and diatoms have a so-called a shell made of silica, which is chemically similar to glass. And if you look carefully, then you can see that there is a pattern in some of the shells here, like a striped pattern. And this is very typical of diatoms, and this is also sometimes used to also test the quality of certain microscope objectives, if you're able to see those fine details in the diatom shells. Okay. So why most of the protists are not pathogens like bacteria, viruses, fungi? Well, I will be honest with you. Most bacteria and viruses and fungi are also not pathogenic. It's only a very small percentage that are medically relevant. Yeah? So a very small percentage. As a matter of fact, there are countless different species of bacteria and microorganisms living on our body and in our body. But they're either symbionts, that means they're beneficial to us, or they're completely neutral. Yeah? We only hear a lot about bacteria in the media and viruses and fungi causing diseases because that's, when they cause diseases, that's why they're a problem, right? And that's why we know about them. But if you just look at the biodiversity itself, the vast majority of them are completely neutral or they don't do anything. Yeah. Yeah, so where is something wrong with the audio, or just me? Is there a problem with the audio, or is the audio too loud, or too? I'm going down right now a little bit. Okay, audio good. I went down with the audio level now a little bit because I've seen it for whatever reason it was very loud before. Okay. Audio is good, but it did stutter a bit. Maybe there was some internet problem. Okay. I left and came back to fix audio. Okay. So I hope that you're able to hear me properly. Okay. Well, yeah. So this is kind of the thing here. How to know I have a good microscope? You're gonna hate my answer. You have a good microscope if you're satisfied with it. Okay, the best microscope is the one that you're going to use most often. Many people think that you have to spend a lot on equipment, and sure, there are some pretty good microscopes. My microscope is a pretty nice one. However, it's like this, that even basic introductory microscopes like I have over here, okay, are able to produce surprisingly good results. Okay, they might not be quite as convenient to use because they only have one eyepiece and so on, but generally, you have the bottleneck in many cases is not the microscope, but the bottleneck is do you have specimens to look at? Are the specimens properly prepared and so on? So all of these things actually probably influence image quality also quite a bit. Yeah? So this is something that I have to tell you here. Yeah. I did not use agar plates at home, but I wanted to know how to see bacteria of a phone. I have cyanobacteria in my aquarium. Is this something cool to look at under the microscope, or is it boring like other bacteria? I did make some months ago a video on cyanobacteria. Generally, the issue with bacteria is the following. I mean, the cyanobacteria, they come out in colonies, so that might be quite nice. However, because bacteria and cyanobacteria generally are significantly smaller, therefore you cannot look into the cells quite a bit. Okay? Look here's some other algae over here, right? Yeah. So, and this basically means that the cyanobacteria can sometimes be interesting because they're clumped together in colonies. But if you try to look into the cells, you're not going to see a lot, because that's basically generally with bacteria inside, they're homogeneous. Yeah. So I would simply say you try it out. If you've got cyanobacteria, scratch some off from the window, and of the window of the aquarium, scratch some material off, put it under the microscope, and I'm quite sure that you're not only going to find cyanobacteria, but plenty of other things as well. Okay. So the question is this phase contrast? No, this is just regular bright field. Okay. And if I move the filter in, this here is DIC. Differential interference contrast, and if I rotate the prism, yeah, then it will look like this. Okay, so this is now and as I mentioned before, you can get some people wanted to know about this blue background, and yeah, that's basically because of the setup that I have. But what I recommend is I mean, I 3D printed this one myself. Okay. But you can also try to make these filters yourself using some blue plastic or some marker Made some videos on how to do that, and you can get surprisingly similar effects. Okay, so this is just, oh, I just realized I had the wrong setting here on the, okay. See, and that's again, I'm using the 100 times oil immersion objective, and now because the field of view is so small, I mean, I don't have a very good overview. We looked quite a lot at the amoeba before, but now I can't find it anymore. Yeah? So this answers a little bit the question of whether you always need high magnification. You see, that's actually a disadvantage of high magnification. That yeah, I don't have a good orientation anymore. I don't know right now where can I find the interesting things. Yeah? And I cannot go back further down again with to a lower magnification, because otherwise I'm going to damage my other objectives, because they're going to be covered in immersion oil. Okay. What is this here? I don't know if this, okay. Yeah. So, yeah, are there any technical questions that any one of you have? Okay. Otherwise, if you have any recommendations, some of you already wrote in your comments section, you already wrote some of the things that you want to see. Some of you want to see some staining things. I'm gonna prepare something here. What do you think about Olympus OIC monocular microscope? I don't know the OIC, I'm sorry. Okay, but I mean, if it is a brand microscope like Olympus, Nikon, Zeiss, Leica, and so on, then generally these are all very good quality microscopes with some service. And that is, I think, the important thing is also that you have to be satisfied primarily with it. But I do not know the OIC microscope. Sorry about that. Microorganisms are so small, so little radio waves or other waves can essentially cause mutation in them. Yeah, of course. Not because they're small, but radiation of certain frequencies, radioactive radiations, gamma rays, even ultraviolet rays, of course, they can damage DNA. Just like ultraviolet light is able to damage human cells, that's why you get a sunburn. They are, of course, also able to damage microorganisms. This has nothing directly to do with their size. And indeed, that is the reason why in certain laboratories at night they switch on ultraviolet light to disinfect everything. Okay. A second request for a video on which stains for what purposes. This is a complex and long question, because there are hundreds of stains out there. Some of them very, very specific, and some of them very expensive and difficult to obtain. So that's the reason why I rarely use stains, because when I want to observe living water organisms, I cannot stain them, because otherwise the stains would actually kill them, at least many of them. The stains that I generally always recommend is methylene blue. This is a general purpose stain. And I also recommend, if you want to stain starch, of course, iodine. That is a very common one. If you want to stain chromosomes, carmino acetate is but it means basically you need to prepare the specimen. So there are a few highly specific stains. Also, for example, for the certain standardized stains for staining white blood cells and identifying them afterwards. Okay. Simple staining on curd bacteria. So basically, if you just want to stain them, then essentially you can use safranin, which is a red stain, or you can also use methylene blue. And yeah, safranin is okay. Yeah, safranin. Safranin generally stains pretty much a lot of bacteria red. Okay, it's a general purpose stain, but it will not only stain bacteria, probably also other organisms in there. So you have to see whether it doesn't also stain other things that are in your sample, because then you cannot distinguish it anymore. That's actually one of the purposes of staining, is so that you're able to stain only the things that you want to see, and that the other things therefore are invisible. Okay. What do you recommend to view thick plant tissues under the microscope, concave slides or normal flat slides? Honestly, if you want to look at thick plant tissues, then I don't know if any slide is going to work, because it's going to be too thick to allow light to go through in the first place. So this is generally, so thick specimens with compound microscopes are generally a problem. Okay. So this is something that I just also wanted to say. What you can do, however, is you can put a thick plant specimen, like a small leaf or so, which is a few, let's say 2 or 3 millimeters thick, on a slide. Doesn't matter if it's concave or not. And then you use the low power magnification 4 times objective, and you look at it from the top by having a flashlight from the top. Okay, so you just see the surface. This is something which also works with relatively thick specimens, but having light go through a thick plant tissue is not going to work so well. Okay. What is your timetable of microscopy? I do not fully understand the question, unfortunately. Do you mean what my for making the YouTube videos, or yeah? Or in the past, what my basically progress has been? I'm a little bit unclear about this question here. What is your timetable? And hi from Italy. Okay, yes, hello. I think if you just joined, I have to tell you that I'm going to very soon have to stop, because I'm already doing this over an hour. Yeah, but there is another question. I have a question. I usually collect plankton with a plankton net, but I wanted to try to conserve the specimen for long time observations. How can I do that? So if you, the question is now the following. Do you want to preserve it alive? Is this the question? Or do you want to make a permanent slide? Okay. So this is a little bit the question that I have here. If you want to preserve the specimen alive, I'm going to switch over to the desk now. Okay, what you can do is you can of course add some pond water, and you can simply empty the plankton that you collected into a jar, and you always make sure that when water evaporates, you add some more water. So that is of course one possibility. Yeah, and I've kept water samples like this for months, really. There is going to be a natural change in the composition of the microorganisms, depending on temperature and light and oxygen concentration. But I've kept some of them quite for months. And if you want to make a permanent slide of plankton, then you have to use a mounting medium called glycerin gelatin or glycerol jelly. And this mounting medium is water-based, and it does not dry out completely, because many of the plankton water microorganisms are very, very sensitive to water loss. And if you dry them, they're going to shrink up and shrivel up, and they're going to completely lose their shape. So you have to use a permanent mounting medium that is able to preserve them. And that's usually glycerin gelatin. Or if you want to keep a fresh slide, not one of a permanent mount, but of a temporary mount, I have something that I also showed you already last time. What you can do is you can make yourself something like this here. You put some water in the bottom, and then what you do is you put the microscope slide with the cover glass on here and the lid on top. It's called a moist chamber, and then you can preserve also the microorganisms on this microscope slide alive. Okay, so that's also a possibility. Okay. Video on stomata, yes, this is also something that I can do. You can take videos or pictures of stomata by putting some glue on the bottom side of a leaf and allowing the leaf to dry, and then you peel off the glue and you put the glue under the microscope. Then you see the impression of the leaves and also the stomata, the stomates. Are there any protists who live naturally in our body? Why not? I mean, malaria would be one of the bad ones. Many protists that occur in the body, I think they are pathogenic. I mean, mostly they're bacteria, but I wouldn't be surprised. I think I actually once put some saliva under the microscope and I also saw some protists there. Yeah. It's more simple to study them is plankton from the sea. Yeah. Yeah, then you use basically put them into a jar with some seawater. Yeah, like a small aquarium. I also read some substances added to water to preserve the entire sample. Yeah, you know what this means? Preserving a sample, you got to be careful. Not microorganisms. If you add some preservative to the water, then this is added to kill off bacteria and microorganisms, so that whatever sample of plant material or whatever you have or tissue will not turn bad. But those substances are actually designed to kill off microorganisms, because the microorganisms break them down. So this is actually probably this material that's added, the substance that's added is there to preserve the tissue, that you want to observe, but it's going to kill off microorganisms. Okay. My timetable as a microscopist. Okay, I think I'm going to do the following. I'm going to make a separate video of this. Well, but a very short answer. I started microscopy, well, quite a long time ago. I would say maybe 25, 30 years ago during university time. Since 2018, I've been making YouTube videos. That's why the YouTube channel was opened about 4 years ago, 5 years ago. Basically and recently I started to do some live streams. Yeah. Do you have any video about careers involving microscopy? I did make in this very channel some years ago a video about the use of microscopes generally in research, because somebody wanted to study microbiology because of the use of microscopes, and I said, well, actually I did not use microscopes that often during university time because it was mostly biochemical, but it depends a lot on what you actually want to research. So this is so I'll think about this as well. Thank you, by the way, for the comments, because they are important questions that I can actually pick up on in a later video. I've seen some microscopy videos where they recommend dissecting needle to lower the cover glass rather than dropping it. Do you ever do this? Well, the thing is the following. I don't do that. You can do that. Essentially, what you can also do is you can use I'll do it for the sake of simplicity, what I always do. Okay, it's a little bit too far away. I have to lower this here. Okay. What I usually do is I take a cover glass and with my fingers here on the side, and I simply lower it like this. You can also, of course, use tweezers to do that. Okay. If you want to. I think it doesn't really matter so much as long as you minimize the air bubbles, but sometimes even a few air bubbles are okay. Yeah. So whatever is most convenient for you. Yeah. Where can I find those substances? I usually fluorescents at home, and some copepods aren't very collaborative. You mean basically the substances for preserving the water samples? Essentially, if you want to kill off microorganisms, you add a little bit of alcohol. Okay, what's going to happen is it's going to kill off. I've kept some plant material, but then you're going to kill off pretty much everything in there, right? But I've kept some plant material dead, for almost 20 years in a jar of alcohol. So this is actually able to store quite well, but don't expect to be able to see live things then. But essentially, these are then simply specimens that you keep for a long time and have them preserved, right? So when you got my lab pictures, okay, I'm going to check this again. I thought I showed some of them already last week, all that I got. Okay. You know what I'll do is then I will try to get everything again. Honestly, I don't. Yeah, I checked again today on Instagram, and I've all of the pictures that I've got. I already showed them, so I have to see if for whatever reason I overlooked something. Okay, Happy New Year. Is large 38mm tube a cell phone adapter? Don't find it. Maybe you can tell me what parts should I look for to be able to make the most of it. It is a large 38mm tube. A cell phone adapter don't fit. You've got so basically a large. There are certain cell phone adapters out there. You got to check again one of my videos and previous ones, which can actually accept very large diameter eyepieces. Okay, I know those are some of those are sometimes not very, the ones that I showed you, yeah, might not be large enough, okay, but there are some adapters out there, or you have to make a do-it-yourself solution. Yeah, so that's a little bit the problem. Hello from Michigan. Have you used plastic cover slips? No, I have not, because the plastic cover slips generally, they do not, for me, they don't give a very good result, and I also use polarized light, and for this reason they disturb the polarization a little bit. Okay. But what I have done in the past, also again not very good with polarized light, is I've sometimes used tape. So if you use sticky tape, you can collect some skin cells, some face mites maybe, and then stick that on a microscope slide, and then this will actually also work for some specimens. Yeah. I use a homemade camera adapter for my Olympus PHP. I used simple 10x oculars and some parts to connect. Yes. Unfortunately, fortunately, sometimes it's necessary to go to homemade solution. Okay. The alcohol doesn't change the shape of some. Yes, it does. Alcohol, if it's concentrated enough, will remove the water of the microorganisms. It will kill them, and it will actually also shrink them, because water is lost. So what some people are doing is they will add a little bit of vinegar, acetate. Because apparently acetic acid will again cause swelling to compensate the loss of water by alcohol. So there are certain mixtures, they're called fixing mixtures. You fix the specimens this way, where the alcohol will remove the water and it will preserve it, but the acetate that you add, acetic acid, that's like in vinegar, will cause the microorganisms to swell again, so that their original shape is preserved. So there are certain solutions that actually allow you to do that. Okay, can you use this time slot for your next coming live streams? Okay, okay, I don't know. Usually this time is not very convenient for me. I know it's more convenient for other people. Maybe some kind of a compromise in the future. I don't know yet. Yeah, the reason why I'm using this time slot today is because of an appointment, and that's why I have to stop very soon. Yeah. Why only few people know about microbiology or microscopy? It's a very interesting hobby. Yeah, that is correct. Amateur astronomy, which I've also been doing, is more popular for whatever reason. Okay, it's got more coverage. And I think one of the problems could be is that microbiology, microscopy and so on is not quite present in the media quite as often. Usually it's always present in the context of diseases, medicine, okay, bacteria which are dangerous, you know, and astronomy is a little bit more popular. Space travel, okay, rockets, and so on. Yeah, and for this reason, I think this also contributes a little bit. Can different microscope head fit with other microscopes? Generally not. Be careful. Yeah. Kind of like Omax head with Amscope body. In the case of Omax and Amscope, it depends on the individual model, and Omax is an Amscope brand, if I'm correctly informed. So it really depends a lot on the microscope. Yeah, but generally you cannot assume that this is going to work. You have to look individually. Yeah, watching from Greece. Yes, hello, and I am going to buy MO2 microscope. It is good. It provides magnification up to 1500 and costs $100 in India. Okay. A microorganism small star is huge. Yeah, that's and in both cases we need technical devices to view them. Please review Indian microscope brands also. I don't have, honestly, I don't have access to those. But often it is like this, that sometimes the microscopes are very similar in any case. Sometimes they're manufactured in China and then sold in different parts of the world using different brand names. Usually that is the case. Okay. Okay, thank you again here. What do you think about magnification changer? In what sense now? I have to see this. I mean, you mean this is the way that I change magnification. However, see now the lamp. Okay. I mean, there are certain, I know occasionally some microscopes allow you to change the magnification at the eyepiece. I mean, you can do that, but usually this does not improve the quality. Okay, usually the quality is worse. Yeah? I showed my family rotifer in the microscope, and they were wondering what kind of alien it is. Yes. How can I see smoke or diffuse materials under the microscope? Very difficult. You can see smoke particles by allowing the smoke to deposit on a microscope slide. Okay, that's the only way. You hold the microscope slide into smoke and hope that some of the smoke particles will settle on it. For some microscopes like the BHP, there are a device that is put between the microscope body and the head. Ah, okay, I see. Never tried those. Yeah, I heard about those. Where basically you put something between, to use this here, some kind of something in between here. I've heard about that. Now that I remember, but I have never used those. Generally, I would always say the more that you add in between into the light path, any optical system will only decrease the contrast. So the question is if it is really worth it. You'd have to try. Okay. Are those small green? The small green one? No, the small green ones that you see over there are probably diatoms. Okay, now I've not completely lost everything here. Yeah. Folks, I think I slowly have to call it quits because I have to go somewhere. Okay, so because the spirilla that you basically, the spirilla that sometimes are present, they are bacteria, and they are much smaller. Okay, folks, I have to call it quits for now because I've been now talking already 1.5 hours. I'll think about the time again a little bit. What is the most convenient time? And I think I'm just going to stop. Why don't you put fluorescent on your microscope? Working on it, not this specific microscope, but I'm trying to experiment with blue LEDs, blue LEDs, where you can do some basic fluorescent microscopy using algae, where the chloroplasts start to shine red. So that's called autofluorescence. You can do that. It is a little bit experimental and still working on it. I have not figured it out completely yet, because you have to get the right wavelength as well, but it is possible to do basic fluorescent microscopy as well. Even homemade solutions are possible. You have to get ultraviolet UV LEDs, and then you can do some experimentation there. Okay, so I'll leave it at that, and I wish you all the best. Okay, I gotta quit now. Happy microbe hunting, and I'll be online again hopefully next Saturday. Still a little bit uncertain about the time, but probably I'll go back to my original time that it was before. Okay, all the best. Okay, I'll remember that my objective is in the oil. That's really important. Okay, so let's lower this right away, and that's going to be and then I have to find some tissue, some lens paper, and then I'll clean it off. Bye bye. Happy microbe hunting, everyone, and see you around. Bye bye.